welcome to the research roundtable. We've been doing these for a while, kind of just internal to our team, and having a presentation and discussion. This is the first time we're having some people listen in so they can see how these science discussions work and be part of the process. So we're going to be having a presentation today by Emily, and I say David and Tatiana are here from ImYoo, and they're an innovative company that does single cell immune profiling with at-home collection. So I came across them when I'm part of the patient-led research collaborative too, and I came across them through that and just thought that they were doing really cool stuff and that it could be really powerful for at-home studies of ME/CFS and long COVID, and particularly for severe and very severe patients who can't participate in a lot of clinical trials. So yeah, I was excited to work with them. And last year in March, we did the pilot study and collected multiple samples before, during, and after the antibiotics. And they'll go into more details about that and show some of the results. But I think we've been learning some interesting stuff. So it'll be a fun discussion here. People might recognize some names and faces here. We have some people from the Renegade research team who I refer to as our science geeks, who really like digging into all the research. So we have Sam, Nick, Rochelle, Isabel. And then thank you to some of the researchers that have joined here for the discussion, Rob, Andrew, and Carrie. And we chatted with all of you kind of early on in the Remission Biome process. So it's fun to circle back around now when we have some results to discuss. So yeah, it looks like it's settled down with people coming in now. We might have a few people join here in the next few minutes, but I'll let Emily go ahead and start presenting.
Sure. All right, time to share the screen. And we'll pick this desktop. Oh wait, can we do the window? Advanced portion of screen. There we are. Okay, so you guys can see this first page here, right? Sort of summing up everything.
Yeah.
Incredible.
See the whole browser.
Great. So let's get started. Hi, everybody. A little bit more about ImYoo. What we do is we generate a molecular snapshot of your immune system. And in doing this, we can generate big molecular data from home. For the purpose of the Remission Biome study, we were really excited to potentially capture a shift in gene expression in Tess's immune system. Why are we looking at RNA at ImYoo as opposed to DNA? You can think of 23andMe, that could tell you about your heritage, which is cool, but that's pretty static. It doesn't change. What RNA can show us is how your genes are dynamic and expressing more or less at different times of day, or in reaction to different things. So that's what we wanted to do, was we wanted to see what genes were turning on and off in Tess's immune system. And we call that gene expression. She had this really cool idea that sort of, from my understanding of it, melded together these two hypotheses about ME/CFS. And definitely, Dr. Phair, Dr. Grimson, or any person in the call, correct me where my understanding of the theories are not accurate. But from my learnings of this, it looks like there are some hypotheses that get at ME/CFS as a vicious feedback loop, or a couple vicious feedback loops. And there's this neuroinflammation hypothesis that nerve cells are inflamed. And then there's this metabolic trap hypothesis that I know Dr. Phair has really paved the way in developing, where that feedback loop is in the mitochondria, those energy factories of the cell. And there is this idea that these may be interplaying with each other and driving each other. So Tess had this incredible idea based on her own personal experience with antibiotics in the past. Could she trigger this remission event, an alleviation of her symptoms? And can she do that through the microbiome by taking amoxiclav antibiotics and a series of other supplements and interrupt that cycle? So she came to us with that plan. It sounded awesome, and we were just excited to observe it and capture that on a molecular level, not just symptomology. So we set out to capture 9 snapshots. Now, keep in mind at the outset, we didn't know if Tess didn't know she would go into this event or not. So what's cool about ImYoo is, we just send over our kits, and if it happens, we can capture it. And that was the opportunity we presented for this N of 1 study. And I think that, luckily Tess did experience some remission events, and we got some pretty exciting results to share from these 9 molecular snapshots. So I'll hand it over to David for the next slide.
Yeah, instead of me switching to, can I just talk off of your slides? Okay. So what we get when we do our lab work, so we isolate the white blood cells from the sample, and then we use this company called 10x Genomics. They have a lot of competitors out there too, but they do these assays called single cell assays, and what that means is that we measure things for each cell separately from each other. In the case of 10x Genomics, they do it by putting each cell in a little water droplet that's separated by oil. It looks really pretty. But the end result is the same. You get data on each cell. And so the picture you're looking at here is what's called a t-SNE plot. The fancy description is it's a 2D projection of cells in a high dimensional space. And it's kind of like when you take a globe and you flatten it into 2 dimensions. It creates some things that are wrong, but it's a way to visualize something on a piece of paper. And in the gene expression case, the genes are in a 30,000 dimensional space. And what that means from an interpretation perspective is when you look at this plot, every dot here is a separate cell that was one of the cells in this experiment. And how close the cells are to each other is representative of how similar those cells are to each other. So similar cells grouped together. And a big caveat, t-SNE is not intended to be interpreted directly. It is a visual aid for double checking your analysis and just like seeing what the thing looks like, because it gets boring looking at just like numbers and A, T, C's, and G's all the time. So what we can do once we have a bunch of cells that have gene expression that's similar to each other is we can identify what type and state each cell is in. And so if you zoom in on the bottom part, Emily, here's an example of, we can color the same plot by the expression levels of each cell. And so down here, you see the same plot, but now it's colored by CD14 expression. So how much CD14 is turned on in each of the cells in the plot. And you can see that almost all of the cells that express CD14 are down at the bottom, kind of grouped together. And we can use this to help identify for each cell in our data set what type of cell it is. For those of you that know your immune cells, CD14 is uniquely expressed by CD14 monocytes, and so we know that that group of cells is now CD14 monocytes. And so what we can now do once we have identified the type and state of each cell is we can go do analysis on, I see a question from the panel, looks at white cells only. Yeah, for this experiment, only white blood cells. Sometimes red blood cells sneak in, but we usually don't look at them. So you can now go into each of these cell types and you can ask what genes are different in different conditions. So in this particular experiment, there was a baseline and there was an event, and we had 3 samples from each. And so we started looking at, for every one of these cell types, CD14 monocytes, T cells, dendritic cells, what is different during the event versus what was at baseline. And we have, if you want to go to the next one, Emily. Yeah. And so something immediately popped out when we looked at the data from this experiment, which was the only cell type that where we detected any statistically significant gene shifts was in CD14 monocytes. That's why I picked that one to show you. That was a little bit of a, yeah, I showed that before I should have, I guess. But, and we found, at least at the time of this writing, 21 genes that were significantly downregulated during the event relative to the baseline. That was also another curious thing. They were all downregulation as opposed to upregulation. And from Emily will talk more about this, but from, I believe also from your work, Andrew, you guys also identified CD14 monocytes as a cell type that was strongly affected in, I think it was post-exertional malaise in folks with, after exercise. And this data is incredibly rich. You can like keep going down level after level. So the next level that you can go in is not just what cell types are different, but in every cell type, what specific gene is different. And so if you look at this volcano plot down at the bottom, they're called a volcano plot because they kind of like, the dots like spew out to kind of like the plumes of a volcano. In this case, every dot on here is a different gene, not a cell. So we're looking here at all of the genes that we detected in CD14 monocytes and how different they are in the event versus the baseline. So it's a quick way to visualize kind of like where are the interesting genes. And this is also where we can visualize what things are above some statistical significance threshold. And so everything above a certain line, like everything that spews far enough away from the volcano, is above some statistical threshold and is, a good starting point for where to look. And one of these genes that we found, the one at the very top, is CCL4, and Emily will talk more about that.
Yeah. So we'll hop on down here to talk about CCL4. So CCL4 is a pretty cool gene because it's been associated with chronic systemic inflammation, not just in ME/CFS, but in other conditions. They've seen it in Lyme disease, I believe. I think they've even seen it in obesity. So it definitely plays a role in a lot of conditions. And I wanted to point out, and this will probably come up in discussion, but it's really cool that Dr. Grimson's paper looked at kind of the reverse of what we did, inducing post-exertional malaise, like triggering symptoms with exercise. And we can see that when we compare ImYoo's findings, we do see that inverse, that flipped relationship there. So those CCL4 genes went up when the symptoms were exacerbated, and Tess accomplished the opposite, an alleviation of her symptoms, and then we saw, boop, it goes back down. So it's cool to see this seesaw effect. That was from bulk sequencing, and ours was from single cell. And so what's also interesting is that we saw CCL4 continue to go down in the months following these remission events. So that was really exciting. Also, we know CCL4 in some animal models has been shown to reprogram metabolism in immune cells. Links to those papers and things will be in the article we put out about this. So that just gets us wondering, well, why aren't there human studies to look at that? To reprogram the metabolism of an immune cell really would get us thinking more about this metabolic trap. And I would love to discuss with Dr. Phair after the presentation what ideas that sparks for him. Of course, this is an N of 1 study, and we want patient scientists to be armed with the vocabulary to think objectively and critically about results like these. So I'm going to hand it off to David to get into that a little bit.
Yeah, so I just want to situate this a little bit into this whole finding into context for potential biases. Because I think one of the most important things that I learned throughout like my scientific training is whenever you see something in data, it's always really important to ask the question of what else could have caused what I am seeing other than the thing that I want to see. So just to be like super transparent, like this kind of finding is exactly what we wanted to see from this experiment. When I sat down to go, we've been waiting for this data for like a year because we had to like get a grant and like it took forever. And so here I am sitting down to do this analysis, and I'm very excited and I want to see something. And I'm part of ImYoo. We're a company. We need to, make money to pay the bills. So it helps for me to say that I find things as a bioinformatician. And so there's all these biases going on in my head when I'm looking at this data. And it's very easy to convince yourself that you have seen something that you want to see. There are hundreds of differential expression methods for single cell data. So this particular method that we're using is one of them. And so you can go down these rabbit holes where you're like, I'm just going to try all the methods until I see what I want to see. And so what I try to do when I'm doing analysis like this is ask this question of, what else could have caused what I'm seeing? What are some other analyses that I can do to make sure that what I'm seeing isn't being triggered by my biases? And I think a good a concrete scenario that just recently happened that kind of illustrates this is the eclipse. So we all saw a lot of us saw the eclipse earlier this week, and I think it's important to remember that thousands of years ago, when people looked up at the sky and saw the eclipse, they had, dozens of different very compelling reasons in their minds of what was going on when they looked up at the sky. Like, I don't know the different mythologies, but I think one of them is like there's a dragon that's eating the sun, and so you have to scare the dragon away, or else the sun will be gone forever. And it took people that were in those time periods and in those cultures that were hearing this is what's happening. It took people to say, you know what, maybe there's something else that could be going on that isn't what everyone thinks, that isn't what I'm supposed to think and what people want me to think. To help us understand new things about how the universe works. And I think it's the same thing here with this, especially with this N of 1 study, that we have to ask ourselves, like, what else could be going on? So I don't know if you wanted to give a few examples of what kind of things could be going on, Emily.
Yeah, well, I mentioned a vocabulary, and as a public benefit corporation, like we have this mission statement to engage people in the process of scientific inquiry. So having a vocabulary to think critically is very helpful, right? To think about disease variability, which in the space of ME/CFS, I feel like that doesn't require much explanation. Everyone here knows that their ME/CFS is different from another person's. It's very unique. So just because this worked for Tess doesn't mean it'll work for someone else necessarily. That's why bigger studies should happen. There could be technical artifacts. So when we separately batch different samples and we process them at different times, there are, technical artifacts that could show up from that. Human error, that's in our common lexicon. We know that there are humans doing the science. David is a human analyzing the data. Jeff is a human in the lab. And we just have to be cognizant of that. And Tess is a human who collected her sample and packaged it and sent it back to us. And confounding variables is another helpful term that we hope to introduce into the vocabulary of our patient scientists so that they can think critically when they collect their sample. Am I experiencing seasonal allergies today? Could that be a confounding variable? thinking about these things, because with longitudinal studies, we are conducting this experiment over a longer period of time that affords us so much more clarity, so many more molecular snapshots, but we also have to think about the other things we're introducing over that window of time.
Emily, I'm going to jump in and add another important one.
Mhm.
This could also just be the signal that you're seeing could just be a normal thing that happens also in healthy people, right? This could just be something that maybe everyone's CCL4 sees this trend before and after antibiotics. So it's a very important one to consider here because we did not have a healthy control taking antibiotic. So that's just another one that I want to add there.
Yeah. Okay. So basically, we did share some of these findings, and with all of that in mind, we hope to get some input from Dr. Phair and Grimson today. Maybe we can add it in to the dialogue. But we spoke to our friend Lavanya at Northwestern doing immunology research in long COVID. We actually met her at the same time we met Tess for that PLRC project. So she was excited by these results because it showed to her that the microbiome could be impacting immune cell activation in Tess's ME/CFS immune system. And again, it brings back these questions about the metabolic trap hypothesis. Is the microbiome an avenue to get through to that feedback loop? And Tess is an expert in ME/CFS. She knows more about the condition than anyone could learn from a textbook because she lives it. And just her testimony of, some people can't leave their house. So the millions missing from every aspect of ME/CFS research, we hope to capture with decentralized studies because they shouldn't be missing from the research. That's the avenue to get them back. So there is maybe one more thing I wanted to highlight, which is, we talked about CCL4 and how in animal studies, we've seen it reprogram immune cells, metabolisms. So that's very much looking at the cellular level. If we zoom in a little closer, though, I wanted to share this pathway that Nick brought to our attention, which is the TLR4 pathway. So another gene hit that we got in the CD14 monocytes, a top hit was this PPM1 gene. And, we'll share this report so you guys can deep dive into the description there. But basically, we know that PPM1 or the PPM gene family can help with producing these membrane proteins that detect something from bacteria, that it tells the immune system, let's fight off this bacterial infection. So the fact that this gene was downregulated, it's maybe not signaling for this protein to mount defenses against a bacterial infection. It does lend itself to other hypotheses that this pathway is heavily involved in ME/CFS. And again, this pathway, it lends itself to that idea that a bacterial infection could trigger ME/CFS, and it's also related to other diseases. It's been tied to Lyme disease as well. So this pathway is very exciting to link these molecular pathways and these downstream effects to a gene further upstream. So these are sort of how we want to contextualize what we found. And in this report, there will be links to, gene tables and the publications cited. But mostly what we're here to do today is to discuss these results. What questions do they inspire? We have some questions for the panelists, and we're just looking forward to people's input.
Thank you, Emily. You can do the virtual hand raising if anybody wants to jump in here.
I'll start with one if that's. Wait, no, there's.
Yeah,
go for it. Go for it.
So this is a great study. Thanks for sharing this. And I think it really sets up as you pointed out, it really sets up a strong motivation to extend this kind of work beyond an N of 1. A couple of technical questions. I'm not really sure who I should be asking this to, but I'll throw them out there. So I think you're really looking at probably what are called classical monocytes, and that's certainly the cell type we saw the strongest signal in. I'm curious, do you see any signal in other subsets of monocytes? And then going back to sort of something you went through right at the start, there's really no signal in any other non-monocyte cells. Is that really what you're seeing? That's amazing if that's true.
Yeah, so I can comment on that. So in terms of no other signal, so for this first passive analysis, I would say the most conservative analysis
Okay.
that we can do. So we took all of the cells of a cell type and collapsed the counts together so that it was like doing a bulk RNA analysis, a pseudo bulk analysis with an N of 3 for the event versus baseline. And I like doing that first because it's like we can be very confident that what we see is real. And so using that very conservative analysis, there were no other genes below the, after multiple comparison correction, because there's 30,000 genes. So you decrease your p-values pretty significantly. After doing that, there wasn't anything else that showed up in any of the other cell types, and also not in the CD16, the non-classical monocytes. However, long story short, if we actually were to do more like single cell level analysis, there's some methods out there that can do that now, like Memento, and there's some packages in R that that aren't so strict. I'm sure there will be other things, but then it becomes more of a now you have to trade off, like, does this fit some story? Is it, maybe you identify a small subset of genes and just focus your analysis on those so that you're not having to do multiple comparison correction across the whole thing. So yeah, I think there's a lot more to do. And
well, so I'll just make one comment along those lines. So it's not really made very explicitly in our paper, but we've done a lot of follow-up work, and other than CD14 monocytes, the cell that has the strongest signal difference when you can compare patients to controls are antigen-experienced CD8s. So if you want to focus on something, and that might be interesting to do.
Okay. Are there any? I haven't heard antigen experienced. Is there another? What else might they be called, or what genes are they uniquely expressing?
Effector or effector memory CD8s.
Oh, okay.
Not naive CD8s.
Oh yeah, I should mention in this analysis also, we only looked at cell types at a broad level, so we only have the 6 that are up there, but we can go deeper, and the memory T cells, effector memory T cells usually have their own cluster.
Yeah, they will, I'm sure. Yep.
So we can look at that. Very cool.
Tess, I had a question.
Go for it.
It's really compelling that your data are similar to what Andrew reported. If we allow that you're going one direction and Andrew's going the other by inducing post-exertional malaise. I wondered if phenotypically you experienced during this remission that PEM was gone.
Yeah, all symptoms gone, and then for the months afterwards, still I was in remission. I didn't push it much. Like I wasn't going to the gym and trying to work out or anything, but my energy envelope definitely went up. Like I was doing a lot more. I was able to work, do a lot more computer work and just be more active in general, and without PEM.
Did you find you could think better?
Mm-hmm. Yeah.
And how long did that last? For
a few months. And then what sort of knocked me back, I got a a herniated disc in my neck. So I started having symptoms from that. And so I'm not sure, if that didn't happen, how long that would have lasted.
Does your whole analysis suggest a mechanism for the 24 or 48-hour delay that we find in PEM?
That's a good question. I'm not sure. Do you have any thoughts on that, Nick?
Yeah, no, it's difficult to know from this analysis. As David was saying, is pretty conservative. We're looking at like 21 genes that are specifically downregulated, and even though these are all in the sort of same, well, the vast majority of them are in the same area, like they're involved in the nuclear factor kappa B mitogen activated protein kinase signaling. A couple of them were to do with glucocorticoid and hypoxia inducible factor regulation. So like yeah, everything's in that area. It's just we don't have enough clarity on all of that yet. Like if we can have a look at the new analysis, I'm pretty sure like once there's more than 21 genes, it's a higher bar that we're setting. If everything's still in that area, yeah, it's a stronger signal. Yeah, well, everything is a reasonable signal as David already talked about previously.
I think Emily was next.
Cool. I do want to, I know I glossed over these like big hypotheses, and I'm curious about the neuroinflammation hypothesis. Do these CD14 monocytes and what we observed in them, would that support the neuroinflammation hypothesis of ME/CFS? I see Andrew kind of. I'm curious to get the input there.
It's hard. I mean, it's hard to answer that. I think it supports. It's consistent with monocytes being important. Does it specifically support this? I'm not sure. I don't. It certainly doesn't reject it. I mean, I think the idea we really proposed in our work was based very much on the basic biology of monocytes, which is if you're inappropriately activating them, what monocytes do is they move from the blood into tissues. And what we're trying to do now is in the next series of studies is to look at that directly in tissues. And there's some other groups as well, also looking at that kind of thing. We're not looking in the nervous system, but there's other groups that are. So I think those are the key studies there. I'm not really giving you a clear answer, but I don't think there is one on this.
I didn't expect a clear answer. It's ME/CFS. It's all over.
I think maybe Nick and Kat have been putting a little bit of thought into this about how this could relate. Does either of you have any comments?
Yeah, and look, going back into what I was saying before, I should have clarified a little bit. This nuclear factor kappa B and the mitogen activated protein kinases, they're activated by a load of different things. So yeah, there's an excellent point put up by, I believe it was Tatiana earlier. So like something like this could be similar to what you'd see if someone has a fever, like they have increased monocyte activation. But in that situation, you'd expect to see changes in more cell types, and like you'd see T cells being activated and neutrophils, and depending on whatever threat, like you'd see different responses. So just seeing the differences in the CD14s is good. The other thing is that if there's a neuroinflammation causing changes to the monocytes, there's got to be a specific mediator of that. So Like there's a few studies in long COVID that have pointed to a high mobility group box protein one, so HMGB1, as something that can do this. There's other things that have been talked about, like substance P, CGRP, and neurogenic inflammation. So yeah, it's gonna have to zoom into different things and see what's important in the future.
Thanks. Rob?
What are you thinking are the chain of events between the antibiotic and the change in CD14, which I think of as a co-receptor for TLR? Is that the way you think of it?
Go ahead, Nick.
Yeah, no, a CD14 is a toll-like receptor for a co-receptor. Yeah, there's a couple of different theories. Cat is probably the best to respond in the glutamate part of it. Yeah, I'll let Cat go. Yeah.
So I've only just started on looking at CD14 in specific, but it does seem to interregulate with some of the transporters that we're looking at for glutamate in the brain. One of my major questions is whether or not the changes to the transporters in the brain affect those transporters systemically, because they are also very, very highly expressed in the gut, and also in the joints. And given the large overlap we have with EDS, I think that that's going to end up being a key factor as well. Sorry, the foster puppies are a little upset with me right now. So in terms of that, I was actually wondering, in terms of what is expressed on white cells that we could track for that, to see if that is part of what is happening, or whether this effect on CD14 is something different, would actually be XCT, if that is trackable. I didn't see that on any of the results here. I'm not sure if that's something that's possible.
So is the question, can we track the downstream effects in other tissues?
So one of the effects that I'm looking at, as far as what we know amoxiclav does in the brain, is the effects on the glutamate system. I've been looking specifically at the glia, at the transporters GLAST and GLT1. Those aren't expressed on white cells, so we wouldn't be able to track that in this way. But the XCT antiporter from the glia is expressed on white cells. And I think that that's going to end up being our next roadblock, for those that do get that first half of the system working again. Making sure that XCT is also functioning correctly will be also key. So I'm wondering if that is something that we can track or not. Because that's been the hard thing, is a lot of times, without having necropsies, it's a little bit hard to track the exact changes in gene expression in the brain. So if we can find any form of surrogate for that, that would be immensely useful.
And just if I'm understanding your question, are you asking about the data with respect to gene expression, just wherever there might be data of it? So when you say XCT, you're looking at the gene expression?
So, yes, because that's the effect that I'm seeing
system
from the amoxiclav is changes in the gene expression for GLT1, GLAST, and XCT. They are all, the expression is definitively changed. But the question is, does that line up with our remission experiences or not? So that if we can actually track that directly, I think that would be just immense.
Yeah, it might be possible that the increased neuroinflammation is just increasing the monocyte migration into the brain, as it is seen in other conditions like multiple sclerosis. So in that case, it could be just a multitude of factors that are increasing chemotaxis. Not like obviously CCL4 has been talked about. Yeah, it increases the chemotaxis of monocytes and macrophages and dendritic cells and other immune cells. So it could be monocytes are being recruited, which recruit more monocytes in that fashion. But yeah, no, we'll have to wait and see.
Emily?
Okay, Rob, so want to dive into the metabolic trap hypothesis that you've been working on? first question, maybe it needs to be broken down a little smaller, but can you modulate metabolism through the microbiome? Like, can you confidently say that?
Absolutely. The whole pathway that you're talking about here is a good example. You stimulate a receptor, which is a receptor for LPS. You generate the typical infection response, which includes both NF-kappa B activation, the way Nick described a few minutes ago, and also the production of type 1 interferons. And as soon as you get type 1 interferons, now you have the possibility of, I don't know, there probably are 300 or 400 interferon stimulated genes. One of which is the one that I focused on, that's ACOD1, which produces a gene product called CAD. And CAD, or cis-aconitate D carboxylase, is the enzyme that creates what we've called a short circuit in the TCA cycle. It converts cis-aconitate to a taconate, which is by itself a metabolic change. This pathway diverges from the classical TCA cycle. And so you're not making as many reducing equivalents as you were before. And the mitochondrion that's affected is suboptimally working. It's making less ATP. And that seems to me to be a clear example of a modulation of metabolism caused by a change in LPS, or presumably also in bacteria.
Great, thank you.
Does that answer your question?
I think so. To contextualize these findings in the context of the metabolic trap hypothesis. So it looks like what we've seen here supports the metabolic trap hypothesis, or if not, what would you need to see from this study to say, yes, this is supporting the metabolic trap hypothesis, or does it not?
Well, as you've mentioned, all of you have mentioned, you're stuck with measuring gene expression in blood immune cells. We all are. It's not like it's just you. That's what we have. And so we're, in a sense, we're doing the classical error of looking where the light is shining. It's like the classic drunk looking for his keys under the streetlight. And we look there because we can look there. Doesn't mean that we're going to find something there. And we're, for the metabolic trap, as you call it, I'm thinking of the words I call the attack-innate shunt. Those things are happening in tissue cells. And so we're forced by the limitation of taking only blood samples to look at immune cells in the blood, which is great if what you want to look at is that part of the immune system that is functioning in blood. But the innate immune system is happening everywhere. And so if you think of the symptoms of ME/CFS as arising from cell types other than immune cells, then you need to be able to look at those other cells, and it's very hard to do in a patient. So the thing I would look for if I had only blood immune cells is evidence that those cells have seen a different environment when they were trafficking through tissues. And that's why I always ask Andrew, did you find any interferon stimulated genes upregulated in any of the cells that you see in the blood? And he says no. So in that sense, we don't find support for the attack on H1 hypothesis looking in the blood. The other point I would make, and I've made this many times, so it won't sound new, is that when we're looking in the blood, we're looking at some linear combination of molecules that were secreted from normal cells and molecules that were secreted from sick cells. And so we think there's only maybe 15% of the cells in the patient's body that are sick. That makes it really hard to see a signal in the blood. I think that's said enough at this point. What do you think, Emily? Is that an answer to your question?
It gives a lot of context. Thank you.
I just thought of Andrew's study where I think you actually separated what you called diseased cells versus the normal class.
Yeah, I was going to ask about that. So what we were able to see was that the CD14 monocytes really partitioned into two sets of cells. And in patients, there were relatively normal cells, but then also these cells that seemed to dysregulate. And those are the ones where CCL4 is so different. So I'm interested, do you see any evidence of the classical monocytes partitioning in this data?
Go ahead, David.
Yeah, I haven't looked yet.
Okay.
You can look at the t-SNE and squint at it the right way until you see two clusters, but to verify that, I would want to do some subclustering of particularly the monocytes and see if it segments. Do you have any particular markers that you think would separate those out?
Yeah, there's a bunch described in the paper that
Great.
are
Okay.
basically the loadings on a PCA for splitting the different cells.
Awesome. Okay, we can check that out.
I just want to go back briefly to something that Nick mentioned, which is trying to identify the factors that are triggering the changes in gene expression here, and to ask a question, which is, do you by any chance still have banked samples, banked PBMCs, essentially replicates of this? And if you did, would you be interested in using 10x's single cell ATAC-seq to look at that? Because I feel like using these kind of technologies, that would be the best way to get at what are the factors binding the DNA that are mediating this dysregulation. And I'll add that we have a large single cell ATAC-seq data set, essentially a parallel to what's in the paper that's been mentioned here, but we're starting to analyze now. So it would be interesting to combine those if that was possible.
I can answer that quickly. Yeah, for most of the samples, we have replicates still in cryo storage. And we've done 10x's ATAC-seq before on the small volume samples, and it worked. It worked fine. So cool. Okay, we could follow up about that. I didn't know how to look at ATAC-seq data. I'll be honest. It was very different from gene expression because it's so sparse and.
Yeah, it's really different. But I think it's to actually find out what's driving these changes in gene expression. It's probably the best option.
Cool.
Do you have another question or comment, David? I think you had your hand up earlier.
Oh, yeah. My question was going to be what markers are there for separating the cells? So I will lower my hand. Okay.
Who else says questions or comments?
I have one last one, but yeah,
Sorry, go on, Emma. I was just wondering, did you by any chance serendipitously capture platelets in this data?
Not that I've seen.
Okay.
You said you had another question, Emily?
I think it's a good wrap up one. So if someone thinks of something else, please bring your question to the top. But it's this, what's next kind of, if we were to design this for a larger cohort, how would you guys do that? And I'm curious how everyone what their ideas would be to scale this up.
I mean, I'll throw out one idea. Some groups, this is not my idea, but some groups are contemplating looking just longitudinally whether or not there's a treatment regime or not, and then just trying to correlate changes in immune cell status with symptoms as patients experience them. The advantage of that is you don't have the added complexity of treatments, whether people are using multiple different treatments, and all so that would be complicated. The disadvantage is you probably would need an awful lot of data and a lot of money to do that study.
But you said people are doing it now?
They're using that idea. I'm not sure that I have no idea if they're using it with single cell profiling, but the idea has been around for a while to just do a lot of longitudinal sampling and have patient data for the symptoms that they're experiencing. I think there could be value in that.
Do you know the name of I would just want to look into this.
Yeah, we can send me an email.
Okay.
And I think the other obvious thing is, which Rob really brought up, is looking beyond circulating immune cells. This is key, but more difficult.
Sam?
Just sort of a driving question, thinking about next steps and more perspectives than short yes/no questions. What are the unanswered questions remaining from each expert in this room? We've got several single cell RNA sequencing efforts at hand, and just one way I envision that sort of the crowdsourcing, democratizing capabilities coming up is putting it into context, sort of the systems biology. What are some unanswered questions in terms of genes that might not make sense, or data that might be needed towards replication or validation? What are some of the unanswered questions? And again, that's more of a perspective that's not easy to just articulate on the spot. It's exactly this in the few minutes we have left, but sort of one sort of perspective to mull over. But happy to hear any thoughts before we wrap up.
I would just re-echo the notion that we really need to make some measurements that aren't in the blood. Yeah, Andrew said the same thing. I'm all for that. I have the impression that tissue sampling, any kind of biopsy, is very difficult for an ME/CFS patient. It's especially those patients with increased sensitivities. It's not an easy thing. And of course, there are parts of the body that you just can't biopsy.
Do you think that blood sampling and tissue sampling are complementary and should be done both in the same study? Or do you think it's more like, this is a blood study, this is a tissue study, we're doing different analyses? How do they exist in the same world if they do?
I think it'd be ideal if they were in the same study, but costs become an issue. I mean, I will say that part of the Cornell Center, a big goal is to look in tissues, but it's going to take several years before we have the data, and we're only looking in one tissue. But that's what we're doing. And to go back to sort of this big question thing, I mean, I think one thing that I've been thinking about recently is and it's really outside my area, but there's all sorts of cell types and genes that are dysregulated. There's a lot of pretty solid molecular data now, but I don't think that there's really particularly solid and convincing data or hypotheses for what's linking the dysregulation across different cell types. And it could well be very complicated. There may not be a single answer, but finding something that explains the disparate changes across the immune and non-immune cells, I think that would be very useful.
We could extend that notion just one step further and go back to PEM at the same time. There's a conviction in the field, I think, and I'd like to have it confirmed or denied, that you can induce PEM either with a cognitive task or with a physical task. And that if you are a patient who has, say, GI symptoms, your cognitive task, a very difficult one, will exacerbate your GI symptoms. And I worry about not knowing, just as Andrew said a moment ago, not knowing how that can be communicated to the gut. And we know there are plenty of neural pathways between the brain and the gut, but somehow you have to just induce the same symptoms that you already have. I have the impression, again, correct me if I'm wrong, that PEM exacerbates the symptoms you have. It does not create new symptoms. And I worry that a systemic signal that connects the brain that's now working harder to symptoms in other parts of the body should affect the other tissues as much as it affects the ones that are currently dysfunctional. There's a paradox here in both the timing, the 24 or 48-hour delay, and in the fact that different patients experience PEM differently, and that difference corresponds to the symptoms that they have getting worse. What is it that discriminates between the symptoms one patient has and the symptoms another has?
I saw a cat raising your hand.
Yeah, it's actually a very, very interesting question, because for me, the one set of symptoms that is very, very typical in PEM that is not very typical otherwise are those flu-like symptoms, which of course we're looking at immune cells. So that it does seem to reason that that is part of the mechanism, but it's not something that I had thought of before. But like the fever, the like sinus swelling, things like that, those all tend to come with PEM, but tend to be very rare or very reduced otherwise.
Nick? Oh, maybe you put your hand.
Yeah, I like to think, well, one of the models I like to use for trying to think about PEM is thinking a pro-resolving response is a dysfunctional. So again, the major one that we're looking at here is the glutamate GLT1 being downregulated and non-functional. But there's multiple dysregulated responses. So normally if people are releasing histamine or adrenaline, and you're also induce a pro-resolving response, usually by merisins and protectins and lipoxins and those are different types of omega-3, omega-6 mediators. So like yeah, that's part potentially of the puzzle, is that there's just not enough of a pro-resolving response to certain things, and that's probably why there's a variance of some people get PEM really quickly because they have no pro-resolving response and they just fall into that hole. And then some people can stave it off for a certain amount of time for whatever reason. Yeah, part of it might be to do with again, like a peripheral left or right shunting as well. So like if people are ill and they're not extracting oxygen for other reasons and for not just a mitochondrial or nutrient intake, things like that. That might be something to do with it as well. Yeah, Rochelle.
Well, for those that know me, you know, I'm going to get into talking about incretins. So with one of the things that would, in my mind, connect some of the aspects of neural inflammation and other symptoms, body-wide symptoms, along with specifically gut and GI symptoms, or that whole network together, would be the idea of the incretins, that a lot of them are secreted within the gut itself, but not only there. And then like the significant role that ones like GLP-1 have in maintaining intracranial pressure and the production of CSF. And like I just saw a thing this morning talking about some of the different GLP agonists are actually able to decrease the likelihood and the overall risks of things like MS and things as well. And the idea that intracranial hypertension, it could be part of the flow or a blocking of the pattern of how CSF is moving properly in the brain, could be also part of the origination of the different sources that people have for how their PEM manifests, or PM. I always say PEM, it's just shorter for me. It manifests for different people because it's impacting different systems, which is increasing different inflammatory markers depending on which system it's part of. And then when you're looking at other things like GIP versus GLP-1 and the risks of like postprandial tachycardia and reactive hypoglycemia, you see that effect in there too, which then affects again the blood flow and the overall effectiveness of moving blood properly throughout the body as well. So I'm probably rambling a bit, but I just haven't seen any mention yet of how like intracranial hypertension and the incretins and the various gliptins that we've been using to try to moderate things like diabetes have actually been having some success in other aspects of some of these possible neural inflammatory disorders that I don't think I've seen that mentioned much in the ME/CFS research literature.
Yeah, so hypothalamic insulin resistance could be a part of the picture here, because again, if there's not enough GLP-1 in certain people, then that could just potentiate the inflammatory response in the hypothalamus or brainstem. So yeah, that could be something that's worth looking into. Another thing that's more important, again, Rochelle was talking about the neuroinflammation side of things and the intracranial pressure. So it could be that increased inflammation actually increases like lymphatic drainage through the cribriform plate. I'll put this paper in the chat if anyone wants to read that. Again, like doxycycline and other things that reduce inflammation, well, specifically VGF signaling, have been associated with, in rare cases, increased intracranial hypertension. That's a potential mechanism. What is, again, I've got to go back to Bob's question earlier. He's talking about seeing things about 24, 48 hours afterwards. Tess, when she was taking the amoxicillin, she didn't see the symptoms for a while because we think that they cycle. Essentially, the beta lactams increase signaling through the cell survival pathway, PI3K, AKT, nuclear factor kappa B, and that increases GLT1 expression. So like your symptoms get worse for a while, and then GLT expression kicks in when it should have before, and then inflammation goes down. So we think that there's a cycling of symptoms due to this. Again, we need to look into this more to prove that.
Yeah, that just made me realize, going back to Emily's question about further study, we mentioned this cycling before, that during the antibiotics, you can go into complete remission, and then like a switch flipping, you go back down to having a lot of symptoms. And so for further study, it would be really interesting to do more samples throughout those time periods, because the ones I collected were only during those remission periods. So I would really like to see how things change when you're cycling. And then the other thing that came up earlier, but just to reiterate, was having the controls who don't have ME/CFS and long COVID who are taking antibiotics. I think that would be key. Anyone else? See Elroy? Oh, I can bring you up. Let's see. Rob, if you want to go, and then I just invited Elroy to come up to ask a question.
So that gets back to the question of the chain of events that leads from amoxiclav to remission. And I imagine that because of the CD14 monocytes and the CCL4 expression data, that you're thinking the chain of events starts with a decreased signal from the gut to the body, and that is the signal that reduces the expression of CCL4. Is that what you're thinking?
I think that's one possibility.
I think it's more likely that Cat's theory, the increase of GLT1 expression in the brain and the reduction of extracellular glutamate is more of it has more of an impact than changing the gut microbiome.
I see. So you're thinking
They interregulate though.
Mm.
And I still that the really, really key question as far as like the root cause of these effects for me is whether or not the effects on GLAS and on GLT1 are systemic. Because given my own experience and given what we've heard, it seems very, very likely that the upregulation we're seeing in the brain is also happening in the gut. And tryptophan and glutamate in the gut interregulate there. And then if that is happening in the gut, and then the glutamate system in the gut and the glutamate system in the brain are also then it all becomes one, rather than two separate things. But it still remains a question whether that effect is actually happening.
Yeah, I've always worried about blood signals being undetectable in this disease. And so what you're postulating here is a systemic signal. And it's disappointing that we as a group of researchers over many years have failed to find that signal. Of course, we're not measuring everything. We might be measuring the wrong things, but a lot of people have been looking. And so that's why I was so keen on the gut-mediated change in bacteria that might be the signal that gets this moving. But I see your point. If it's not the direct effect on bacteria of the antibiotic, but is instead an effect of the antibiotic on metabolism or even endocrinology, then that's a whole different ball of wax.
I do think that for a large subsection of us, at least, it is both. Because that dysregulation within the microbiome will cause the whole system to shut back down. So even if we turn it on, if that dysregulation is there, it's going to shut back down, especially in the presence of excess LPS. That is pretty definitive that will shut those systems down. So for at least a subsection of us, it's going to have to be both. We have this signal change and the microbiome and the metabolite change. But identifying who actually needs that versus who actually only needs the signal change will really help with reducing overprescription of antibiotics, which of course is something we're always watching.
I'm gonna
Thank you
have
all. I need
I'm
to
gonna
get
oh,
moving.
okay. Thank you so much, Rob.
Yeah. Bye.
Yeah. So I'm gonna have Elroy go, and then I think someone else had raised their hand. Elroy, did you have a question?
Hey, Tess, I'm also going to hop off since
Okay.
it's 11. So
Okay.
thank you.
Yeah, thank you.
I also need to go.
Okay.
But thank you so much for this. It's a really wonderful study,
Yeah.
and thanks to everybody for this, especially
Thank
Tess.
you. Thank you. All right. Elroy, if you want to ask your question still, feel free to come off mute and ask it. But I think David, did you have your hand raised?
Oh, I did. Yeah, I was just gonna say the thing I typed because I realized the chat is only for hosts and
Oh.
panelists. Yeah, so I think there are, as has been pointed out, there are big limitations in looking at blood for a like full mechanistic story of what's happening. And so, I like to think about, okay, well, but what can we do with blood alone? And I think one of the things that can potentially be done with blood alone is identifying what kinds of people would benefit from an antibiotic treatment. So like how many Tess like folks are there? And I think that would be like that's the big open question for me is if you expand this out, you get more people, how many people have upregulated CCL4 that goes down, and can we develop a hopefully like easy and cheap test that could tell you, hey, maybe try antibiotics. Yeah, that would be I guess kind of the path that I see.
Anyone else or some of our team who have been following the chat and Q&A, was there anything that came up that you think is worth asking right now?
One thing I can just say briefly, and it's a longer winded answer and perspective, is that there's some questions, including the nicotine focus and other sort of hypotheses that I think are really valuable to address in the data that we actually have already generated, and start to look at democratizing some of that access to ask questions and analyze them. The long-winded part is that it will take longer to actually get those answers derived. But feel free to voice those questions where I don't know off the top of my head how to answer the nicotine question that we received based on the hypothesis that could be valuable to look at with the overlaps to the data that is significant here. But that's something that I think we should definitely pay attention to and start to look at how we, in this room, would envision democratizing that from actually getting people that might be computer savvy to look at systems biology tools or pieces like that to put it into context and not just look at the people doing the research being a closed off team.
Yeah. Carrie?
I just want to say hello because I haven't spoken yet, and it was all a bit like people chatting about stuff that, well, I kind of did understand because my PhD is in immunology, but they all seem to be getting on with it. But I wanted to say that I think people in the UK would be, because I'm in the UK, we'd be really interested in working out how to do some of this in the UK. We've just done a probiotic study with people with long COVID, and yeah, they'd just be so up for trying this. So we've got the capacity in our lab to do some of the immunology, not the RNA-seq, we'd have to send that away, but the cell stuff we could do. And I wanted to ask Tess, have you got microbiome data that kind of tracks? So how did that track with what you were seeing with the remission event?
Yeah, so we do have microbiome data from Biome site, and there were some changes, but I haven't gotten a chance to really think about how they interrelate. Has anyone else on the team started to dig into that part? We just got these results from ImYoo recently.
Yeah, so we did Biome site with our study as well, and
cool.
similar thing. Yeah, you look at the data and you're like, okay, it's going to take me a bit longer to analyze than I thought.
Yeah, we just got access to Biome cohort analyzer, so gonna start playing around with that.
Same.
But yeah, just in the past few days. Let's see. Elroy, you can give you another chance to come up if you want. And then if not, I invited Troy to be a panelist here so he could mention the nicotine connections that he was putting in the Q&A. If you want to go ahead, Troy.
Hello. Of course, I always want to ask about the connection to nicotine because at least 25% of the people seem to have like remission events with nicotine, and also it's usually very fast. And there's also literature that I sent the link to showing that nicotine reduces inflammation by reducing the expression of CD14. And so maybe in the nicotine study, it might be worth having some people do a similar type of test from ImYoo to see if the same thing happens when they have a remission event. The advantage is that for some people, they just feel so much better on the first day. And then after that, they feel worse when they go through a healing period. And then it kind of depends what happens after that. But you could get maybe some interesting data that could kind of correlate with the remission biome data. And if people are feeling better on different treatments, they're affecting the cells in the same way, that might give more weight to those results.
Yeah, that would be really interesting. And if they're not, if we see like different results, then that could also be interesting. Like David was mentioning the possibility of, figuring out who the antibiotics might work for. So maybe it's different for who the nicotine will help. Rochelle?
Yes, for the nicotine, I mean, I've been able to have 2 separate remission events on nicotine at different times, and functioning actually decreased one time after having a bacterial infection with a kidney infection that happened. And so then nicotine was no longer working. And then just this week, I ended up accidentally triggering, didn't realize I was doing it. I fell asleep with my nicotine patch on and ended up with a massive remission event, like the whole psychedelic experience and everything too. And like just the extreme level of it. So it's one of those, if we want to sometime, I mean, maybe we can trigger me to get sick again and then trigger me to do nicotine and test it and just be like clinical level and see. It'd be really interesting to see how those ImYoo results would go. And I'm in a functional level in which I could probably do tissue samples. So that would also be kind of fun to be like, just let's get as much information out of me as we can. I mean, if I can trigger this, I want to be able to see what I can use to help with it. But yeah, that would just be kind of like, to be able to pull these together to see where and why some of these things happen. So that would just be fascinating. So.
Yeah, but I don't think you have to trigger yourself getting sick.
Well, that happens whether or not I intend to. It's too easy to, it's not a cure. So yeah, I know I'll go down again, but it'd be interesting next time I go down to be like, all right, let's see if we can start to gather some stuff and make this be at a interesting place for science. So
yeah. Nick, I think I saw you raise your hand.
Yeah, there's one problem with looking at what nicotine does to immune cells. It increases vagal tone, so it's going to increase the vagal anti-inflammatory reflex, right? So you're going to see a sort of the impact of that, as well as potentially the impact of reducing sort of neuroinflammation and things like that. So it'll be difficult to differentiate one from the other unless you exactly know what you're looking for. So yeah, we'd have to think a lot about that. For sure, for sure.
Do you think anything would come up in the HRV, in the polar that we've been playing with? As far as
Oh,
what was that?
sorry, you finished what you were gonna say.
Oh yeah, I was just gonna say as far as the vagal tone, would it pick up on anything?
HRV, yeah, I'm not sure if it's the best tool to look for things like after and post events. Like it's useful, like things like the PNM50 for the parasympathetic, like that might be good, but like we need a lot more data before we start implementing that. Yeah, I think these kind of things, the single cell profiling, like this is where we get the most in-depth data. Yeah.
Anyone else? All right. Well, this was awesome. Thank you for everybody coming. And I think we got some interesting discussion here and a lot of things to follow up on. So I think this was successful. This is what we wanted to happen. So thank everybody.
Definitely.
Thank you. And
yeah, thank you for organizing, Tess.
Is Emily still here?
Thank you.
I was just gonna say, when are you shooting for the article?
Well, I was hoping to incorporate some quotes from today's call. So I will follow up with Rob and Andrew just to make sure they're okay with doing that. Maybe a week, or just to be conservative. I don't know when they'll get back to me, and they're busy, I'm sure.
Also, we just got new data.
Oh?
Yeah. So we sequenced everything deeper from complete genomics, and there's the same story is still there, but there's more genes. So we might.
Is there XCT?
Yeah, I don't know. I haven't looked
the
yet.
actual gene name on it.
It's SLC7A11. Yeah, I looked for it. Yeah, it's way down, and the bottom is like 40,000 or something. Like,
okay.
Yeah.
It is minorly expressed even normally, so it would be a matter of looking at if there is a change or not.
But there is a change.
Do you know how many genes are in the new data set, or even the one that you've started to look at the pseudo bulk, just the total gene count?
This is like I literally did this morning, so don't hold me to it. But yeah, 48 now for the CD14 monocytes after deeper sequencing, and there's one other cell type now that has a few genes.
Do you know how many genes it was before statistics? Just total genes identified?
Oh, you mean before you?
Before comparing samples, just how many are in the total data set?
Oh, the number of genes, on order like 10,000.
So XCT is most likely in there. If it's rarely ever recovered, little different chances, but it's pretty unbiased to be able to do single cell RNA sequencing. You get most of what's expressed. So either it's not there or it's one of the many unbiased things you can measure. It's my understanding.
Okay. Yeah, we can look for it. Cool.
Awesome. Well, that's exciting.
Yeah, so I say that because we might have to make a few updates to the article.
Yeah.
For that, but
as far as XCT, so I'll send you the study I was looking at, because it is not it's almost undetectable in healthy cells and healthy white cells, but there are certain cases that cause it to become highly expressed. So there becomes this change. So you may have it may be near nil in some samples, and then if we look and see and there is a change, that would be very useful.
Interesting. Okay.
Yeah, I'll send you the study of that.
Yeah, please. Thank you.
Awesome. Well, I guess I'll close it out and say bye, everyone. Thanks again.
Thanks, everyone.
Bye.
Bye-bye.
Thanks.